Good afternoon, ladies and gentlemen. Welcome to the third annual Professor Barrie Hanchard Memorial Lecture. We are pleased that in the midst of all that is happening around us, we have been able to once again organize an evening of intellectual stimulation the life and work of Professor Hanchard. We're also grateful that Roche Pharma and Roche Diagnostics were able to partner with us for this venture. Our guest lecturer today, Dr. Rhonda Graham, is a medical graduate of the University of the West Indies at Mona and was one of the many mentees of Professor Hanchard. We are all, I'm sure, looking forward to hearing about the lessons in life and liver pathology that he received from Professor Hanchard. I anticipate an informative and exciting evening. And without further ado, I now hand you over to our capable moderator for this evening, Dr. Karen Bishop, lecturer in the Department of Pathology at UWI-Mona. Dr. Bishop?
Okay, good evening, everyone, or good afternoon. Welcome to everyone. Let me again specially welcome Dr. Sonia Thomas, wife of Professor Hanchard, Dr. Neil Hanchard, son of Professor Hanchard, and the rest of the Hanchard and Thomas families. We also would like to welcome the rest of you, including some of our special guests, which includes Dr. Carl Bruce, who at the moment is our, medical chief of staff. We're going to start the proceedings today with a prayer. This will be done by Dr. Suzanne Shirley, senior lecturer in the Department of Pathology here, and also the director of the residency program in anatomical pathology. Dr. Shirley.
Thank you, Dr. Bishop. Good evening, everyone. And I invite us now to enter into a short time of prayer. Gracious God, we thank you for this opportunity that we are able to gather like this and be able to commemorate such a special event together. We are still, though, in the grip of the COVID pandemic in so many ways. So many lives have been affected directly and indirectly. And as colleagues and friends together, I invite us to pause for a moment of silence to remember those who have been ravaged by this pandemic in so many ways. Gracious God, we thank you for the life and legacy of Prof. Hanchard, for so many stalwarts of our institution who have taught us so well. And may the lessons that we have learned, including those lessons that Dr. Graham will share today, continue to inspire and inform an effective response, gracious God, to not just the COVID, but the various challenges that face us, and that we can inspire and inform and encourage the next generation of doctors to continue to hold up the light in a very special way. So bless these proceedings, all who will present, all who are present, and that we may leave this place knowing that all is well in your hands. Thank you, Lord. Amen.
Thank you, Dr. Shirley. Okay, so let me just give you an idea of the proceedings. We will start with some greetings from Professor Joseph Plummer, Dr. Carl Bruce, and Dr. Maria Hosbor from AstraZeneca. So I'll introduce each of them, and then Dr. Neil Hanchard will introduce our guest speaker, Dr. Rondell Graham. So I'm going to ask you if you have any questions related to the guest lecture, to the memorial lecture. Please put those in the Q&A box. Okay, we'd like to leave the comments box merely for comments you might want to make about, things like how you're enjoying the proceedings, etc. And all questions in the Q&A box, please. Okay, so with that, we will start first. We will have greetings from Professor Joseph Plummer. I'm so sorry, I think I said AstraZeneca. COVID has obviously fried my brain. My apologies to Dr. Hosbor, who is with us from Roche, who is our main sponsor this evening. So my huge apologies for that. Okay, so first we have greetings from Professor Joseph Plummer, head of Department of Surgery, Radiology, Anesthesia, and Intensive Care here at UWI and UHWI. He is a senior surgeon who is well loved and respected by so many of us. Also, someone who was very close to Prof. Hanchard. So Prof. Plummer, let me hand over to you.
Thank you, Dr. Bishop. And let me use the opportunity to thank you all for attending this third memorial Professor Barrie Hanchard lecture. So on behalf of the Department of Surgery, Radiology, Anesthesia, Intensive Care, and Emergency Medicine, it is in fact my pleasure to bring greetings to the third annual Professor Barrie Hanchard Memorial Lecture. It is also an honor to bring greetings for various reasons. The first really being as Dr. Bishop mentioned, the personal relationship I had with Professor Hanchard. And I'd say not only I, but most surgeons on staff at this hospital at the consultant level, and I dare say throughout the island, and in fact, perhaps even the wider Caribbean, through his teaching of us, first at our weekly pathology seminars as residents, and then later the close working relationship through surgical pathology. We're all taught by Professor Barrie Hanchard. He mentored us, he told us stories, and we all admired him. And importantly, he also treated us as colleagues. His sudden passing left big shoes to fill. Fortunately, his legacy lives on, not only in the set of fine pathologists that he has trained and who are ensuring that we continue his high standard that we have all been accustomed to, both at the university hospital and in our DM training program, but also through this lecture, which really marks the start of the clinical pathology conference series and the start of the new academic year. In fact, the CPC is one of the many brainchilds of Professor Hanchard. And most of us will recall, as medical students, he would say to us, if we attend every CPC for the 3 clinical years, we do not have to read any books. We'll surely do well in the MBBS exams. Dr. Shirley mentioned the COVID pandemic. And in a year as this was, or is, a year in which we lost another of our DM training stalwart, Dr. Cyril Fletcher, who is a contemporary of Professor Hanchard. It's in fact a pleasure to see that this year's guest speaker, Dr. Rondell Graham, is not only a mentee of Professor Hanchard, but I'll go as far to say that he really has been a disciple of Professor Hanchard. And he has continued the succession line of being the best liver and GI pathologist in the Western Hemisphere, taking this directly from, handed from Professor Hanchard now onto Rondell Graham. I'm sure with this succession, both Dr. Cyril Fletcher and Professor Barrie Hanchard are looking down proudly on the quality academics produced from this noble institution. We look forward to the lessons to be shared by Dr. Graham. So having said these few words, on behalf of Seradic and the wider FMS family, I extend to you all a warm welcome. Thank you.
Thank you, Prof. Plummer. Next, we'll have some greetings from Dr. Carl Bruce, Medical Chief of Staff at this hospital, University Hospital of the West Indies, and also senior neurosurgeon here. And as we said, yet another of us who was very close to Professor Hanchard for a long time. Dr. Bruce?
Thank you very much, Chair, and thank you for having me this evening on behalf of the University Hospital of the West Indies. I know we're in the midst of a global pandemic, but we're excited and actually delighted to bring greetings and to celebrate the life and times, as I often say, of the great Professor Barrie Hanchard, mentor, friend, senior advisor and confidant, and previously, as you would have heard, teacher, father, grandfather, and world famous professor extraordinaire. I always smile no matter what happens because Professor Hanchard has done so much for us as an institution. Professor Hanchard encouraged me to follow in his footsteps and go to Canada, get an experience, and to return home. And then he taught me how to drive, how to walk, how to run, and yes, even how to operate, even though many will say he was not a surgeon. The UHII, as many of you know, is a clinical laboratory for the Faculty of Medical Sciences of the University of the West Indies, founded in 1948 after the Higher Commission in the colonies recommended in 1945 that we needed to train university standard graduates in the West Indies because of the challenges with medicine they started. And so we have this world famous faculty, unique worldwide, that has produced so many great lecturers and teachers and doctors over the years. I believe the impact of Professor Hanchard on the University Hospital as a teaching institution can only be described as being legendary. And even in his latter days, the great Professor Archibald MacDonald will remind you that when he had challenges getting the new basic medical sciences complex off the ground, which was finally finished in 2013, he had to go to Professor Hanchard, who cleared the way for this marvelous structure to be built. And many will say secondary only to the aqueduct that was constructed in 1758 by Thomas Ellison of Ellison Flat fame. I'm also reminded that as a young leader, when we had challenges in the powerful medical committee and we adjourned to seek advice, that, as I said last year, 3 of the heads would end up in the corridor at room 16 to seek the advice of Professor Hanchard as to how to proceed. Fortunately, as I always say, he never charged a fee. Otherwise, he probably would have been a well-off man with the amount of advice he gave of his time so freely. But Prof taught us about sports, the NFL, the Oilers, and other teams that I never knew existed, despite my claim that I was a sportsman. West Indies cricket and the Ashes. He taught us about art and the legendary Jamaican artist, Barrington Watson. We learned about wines in his lectures. We were educated on cars and vacation spots. He even sent me to Ascot Chang and Tina Cosmo in New York and introduced me to Michael Chin in Bal Harbour. And they would all welcome you as friends because they knew Professor Hanchard. I will also be grateful. Before my wife and I got married, we sat in the living room at Professor Hanchard with Dr. Thomas, just off the golf course in Constant Spring. And we were comforted that despite the challenges that we might face, that we had friends who would freely advise us as to how to navigate and proceed based off their experience and their standing in our own profession. So even as we navigate through this pandemic, I believe we're in good hands. And I believe that we need not worry, because Professor Hench has done for us what many leaders ought to do worldwide for the people who they teach. His legacy as a leader is celebrated by all of us, and we feel confident that the UHII will emerge from this pandemic standing even stronger because of the lessons we'd have learned from Professor Hanchard. I say to Dr. Thomas and Professor Neil Hanchard and the entire family, we thank you for sharing his time with us. Long live this giant of humankind. And as I always say, and I always read, and I always remember from my old country church, those who hope in the Lord, they will renew their strength. They will soar on wings like eagles. They will run and not grow weary. They will walk and not faint. Professor, we will never waver, and we will not tire, and we will not falter, and the University Hospital will not fail. Thank you very much.
Thank you, Dr. Bruce. Ladies and gentlemen, I am just sitting in for a few minutes for our moderator, Dr. Bishop, who's experiencing some technical difficulties at the moment. But thank you so much, Dr. Bruce, for that lovely those lovely greetings that you brought on behalf of the hospital. So I will invite now Dr. Maria Horsberg from Roche to bring greetings on behalf of Roche. Dr. Horsberg.
Thank you very much, Dr. Gibson. My name is Maria Clara Horsberg. I'm a medical oncologist, and I work for Roche, not for AstraZeneca. And on behalf of Roche, I want to say that we are very privileged to be the sponsors of this Professor Barrie Hanchard Memorial Lecture. And listening to Dr. Bruce's words, me personally, I'm humbled to be here. Just a few words about Roche. Roche has a proud history of 125 years of contributing to healthcare through research and development of new diagnostic solutions and medicines to improve significantly the lives of millions of patients. As Dr. Bishop mentioned at the beginning, Roche has two divisions, diagnostics and pharma, but we work together as one Roche, and we share the same purpose. We want patients to receive the best care for their health. This is a complex ambition. We cannot achieve it by simply developing diagnostic tests and innovative medicines. Access to innovation is a multidimensional challenge, and we want to actively collaborate with health systems to find solutions that will have a sustainable positive impact. Now, moving on to this meeting organized by pathologists, I want to take the opportunity to acknowledge the outstanding contribution you make to patients' lives. A precise, reliable diagnosis is the first and probably the most crucial step in the journey of any patient. But it also has a crucial role to screen, to predict, and to monitor a range of different diseases. And now the increasing opportunities of molecular testing, the advent of digital pathology, and the increasing adoption of data, technology, and advanced analytics have brought pathology to the forefront more than ever. Today, pathologists play a key role in enabling what is known as precision oncology, and more widely in the new healthcare paradigms, such as personalized healthcare and value-based healthcare. At Roche, through our DIA and pharma divisions, but acting as one, we are very excited to be a part of this transformation in healthcare. And as I said before, we want to contribute with our innovation, our expertise, with our passion and our dedication to co-create solutions that will benefit patients and healthcare systems in a sustainable way. So in this present and future of collaboration, I thank you again for the honor of letting Roche sponsor this meeting. And I hope that we can continue to work together with the common goal of helping patients. Thank you very much.
Thank you, Dr. Hosber. Okay, next, a very important moment in this evening's proceedings. Dr. Nadia Williams, Senior Lecturer and Consultant Pathologist in the Department of Pathology here, GI pathologist, lymphoproliferative disorders pathologist, mentor to members of staff here, and also someone who worked with Professor Hanchard for many, many years after training under his direction. She will do the biography of Professor Barrie Hanchard. Dr. Williams?
Thank you so much, Dr. Bishop. And if I may, perhaps we can cite your faux pas in the beginning as a complication of the AZ vaccine. So many things are being quoted as being complications of the vaccine. But to get to the meat of the matter, this is an honor for me. Barry used to refer to me as this 2IC, as you indicated. Not that I knew what it was initially, but he's second in command, and I benefited significantly from his knowledge. I also consider him a friend, and he was a friend. I think my class was near and dear to him, and I would like to challenge anyone to disagree with me, because I know he used to come and fit with our class, the class of 1980. Professor Barrie Hanchard, Prof. Hench, or B, as those of us who worked with him had the privilege of calling him, was a graduate of the University College of the West Indies in the medical class of 1967. After pursuing residency training in pathology at the University of Toronto, he joined the staff of the Department of Pathology at the University of the West Indies in 1976. Remember, I told you what class I was in. During the course of his distinguished career, teaching and research were important aspects of his life. He authored numerous papers, but is perhaps best known as the principal investigator for the HDLV1 project funded by the National Institutes of Health in Bethesda, Maryland. This project not only produced landmark discoveries, important public health initiatives, and several local and international accolades, including the 1999 to 2000 UWI Vice Chancellor's Award for Excellence, but also fostered international camaraderie that stretched from Jamaica to Japan and from Ireland to Brazil. As undergraduates, Prof taught many of us participating in this lecture today. He was also a founder of the Doctor of Medicine in Pathology, from which the majority of the pathologists in the English-speaking Caribbean graduated. He was head of the Department of Pathology from 1996 to 2004, and until the time of his death in August 2019, was the director of the Jamaica Cancer Registry and chairman of the weekly clinical pathological conferences of the Faculty of Medical Sciences here at Mona. Barrie was the consummate professional, meticulous, accurate, and dedicated, dedicated to the practice of pathology, dedicated to teaching and to research. For this, he was honored by the government of Jamaica in 2011 with the Order of Distinction at the rank of Commander. He was a lifelong learner and a repository of historical information, especially related to the Faculty of Medical Sciences and UWI. But he also had many passions, including many of which you've already heard: fashion, fast cars, fine wine, photography, and sport. Prof. Hanchard was always available, and I'm going to share my screen if the technology doesn't fail me. I'm not sure if it is. Okay, here we go. I think I have made a mistake. You'll excuse me as I try again, because there is a particular from the beginning. Here we go. I think. I'm not sure if you're seeing the slide I want to show.
Dr. Williams, yes, we can see the slide. It probably just needs to go into slideshow if you want us to see it, see a larger version of it.
Right. I am trying to go into slideshow and it's refusing to go into slideshow, just to provoke me. All right. So are you seeing the slide?
We can see the photo from Japan.
Okay. All right. So this is part of who he was. He traveled. He was part of the HTLV family, and he was always available, always accessible. He was generous and supportive. There was never a time I went to him with anything that I needed donations for that he would not immediately reach into his pocket and contribute. He had a genuine interest in everyone, mentoring many medical students, pathologists, and others beyond the medical fraternity. He was very proud of his family, but even more so of his grandsons, in whom he was well pleased. His legacy will continue in these memorial lectures. Thank you for the opportunity to share some of my thoughts and my sentiments related to Prof. Hanchard.
Thank you, Dr. Williams. So we move into the main part of the program today, our guest lecture or our memorial lecture. But before that, Prof. Plummer brought greetings on behalf of the faculty. What we didn't tell you is that Professor Minerva Thame, Dean of the Faculty of Medical Sciences here, she had a scheduling conflict and so was unavailable, but sent us her best wishes for this evening's proceedings and regrets that she could not be with us. So now I hand you over to a very special person, son of Professor Hanchard, Dr. Neil Hanchard. Dr. Neil Hanchard was previously at Baylor College of Medicine in Houston, Texas, where he practiced molecular and human genetics and also worked in pediatric nutrition. He recently moved to the NIH, National Institute of Health, where he is now in the National Human Genome Research Institute and heads the Childhood Complex Disease Genomics section. So over to you, Dr. Hanchard. It goes without saying that Dr. Hanchard's dad, I have to say this for the younger among us who may not have known Prof as well. We have some recent people to join our group. So Prof always spoke with so much pride of Neil and his academic career, sometimes, other things, his Neil's children, his grandchildren. And we know that he continues to be so proud of you. So Neil, over to you.
Thank you so much, Karen. That's very touching. And I also want to just give a big thank you to the organizing committee for being able to keep this going again for another year. I know that means a lot to myself, to my mother, and indeed to all of Prof. Hanchard's family. So it is on me to have the great honor of introducing today's guest speaker. And he is Dr. Rondell Graham from the Mayo Clinic. He is an associate professor of laboratory medicine and a consultant in anatomic pathology at the world famous Mayo Clinic. Rondell hails from Barbados, where he excelled academically prior to joining the Faculty of Medical Sciences as a medical student at the University of the West Indies. He continued his academic excellence in winning medals at the clinical and the preclinical level, including the pathology and microbiology medal, the medicine medal. He was the Allen Berry Prize winner. He won the overall clinical medal. And then following his graduation, pursued a residency in pathology at the Mayo Clinic, where he continued to make strides. He was named the most outstanding resident at Mayo, and then having completed fellowships in molecular genetics and then surgical pathology and then GI and liver pathology, he was named as faculty at Mayo and subsequently one of the 40 under 40 to watch by the American Society of Clinical Pathologists, quite appropriately. Dr. Graham is a member of every pathology society there is on the earth. He has an extensive editorial and teaching portfolio with mentorship of students and residents, and he is an author and co-author of more than 140 books and journal articles, following closely behind his longtime mentor and great fan, Professor Hanchard. I first met Rondell when I was at the Mayo Clinic as a resident. And on one fine Saturday, I got this email from my father. And if any of you know my father, you will know that the kind of praise and ebullience that is on display here is not something that is utilized on all occasions. And so indeed, Rondell had a very special place, and that he would never have asked me to do this if he didn't have the highest and strongest belief in him. And that was not in any way misplaced. And so after I got this email, I replied to my dad. I said, sure, I'll see what I can do. And it turned out that one of the consultant pediatricians, who was my consultant at the time, was married to the head of clinical pathology. And he was also the residency director for pathology. And so we had worked together on something else. And so I sent him an email. I said, you know, we have this amazing guy coming from the same university, and he's really keen to do pathology, comes highly recommended. And he said, well, we'll definitely have a look, but you know, he gets 350 emails from foreign medical graduates all the time, and there's a pile of 10,000. So, you know, I sort of fed this back to Rondell, and then about a week later, maybe a little bit more than that, I was walking the halls and I was stopped by said residency program director, who said, "Thank you for sending Rondell our way. He looks superb, and we're going to interview him next week." And the rest, as they say, is history. And so the Mayo Clinic continues to thank us for doing them this great favor. And so I'm sure that this afternoon's proceedings will show Rondell and his pedigree in the highest possible light. And I look forward to hearing his talk. So take it away, Rondell.
Thank you so much, Neil, and thanks everyone for the opportunity to be a part of this. This is a tremendous honor for me. I was a graduate in a class of 2006, and I love my time training at the University of West Indies in Jamaica, and it's really a terrific privilege to be here to share with you on this particular occasion. I'm going to share my screen now. And I would say, you know, I'm really pleased about the others who spoke before, because they have, I think, captured some of the important things about Prof. Hanchard, about his work, his persona, and his legacy, because I don't know that I'm capable of managing it all by myself. And so I'm really I'm grateful that they were able to share some aspects of his personality, his contributions, and so on. So as Neil mentioned, I work at the Mayo Clinic here in Minnesota. I've been here for a number of years. I hadn't seen that email before, so it just goes to show you two things. One, there's always things to learn, and two, be careful what you put in an email. So the presenters give me this really awesome opportunity to speak in honor of Prof. Hanchard, and I kind of divided my talk into two parts. A little bit to talk about what I knew of Prof, and recognizing that it'd be almost impossible for me to capture the influence that he had on the field of pathology, the influence that he had in Jamaica, that he had in the Caribbean. And the second part, to talk a little bit about my work, which is influenced a lot by my time in Jamaica, and two things that I think I learned from Prof in particular. And to say that he had a major influence in the field is really an understatement. If I could divide it into three parts, firstly, at the level of Jamaica, this will not cover everything that he accomplished in Jamaica, but just kind of one thing. As a former director of the Jamaica Cancer Society, as you heard, he provided leadership for its cancer registry, and this informed public health policy. Of course, he accomplished many other things too in that context, in the context of Jamaica, but certainly a major accomplishment with major, long-lasting and enduring benefits. At a regional level, when you consider the number of people that he taught at the undergraduate level as part of DM programs, and that these people are from all across the English-speaking Caribbean and are distributed across the Caribbean, you can see at the regional level he's substantially or has substantially contributed to the education, the health, and the well-being of much of the English-speaking Caribbean over an extended period of time. And that's really just a dramatic accomplishment in the context of the region. And if you consider now the context of the world, what his work has contributed to in terms of the discovery of the HTLV1 virus and our understanding of its biology and the welfare of those who are affected, you can see just looking at the foci of HTLV1 affected in the Caribbean, Brazil, much of sub-Saharan Africa, Japan, and this research, of course, is influential in the US and in Europe. You can see that this was a person who was not just successful in a national context, not just important in a regional context, but truly was influential globally. And I think the others have shown you that despite a tremendous treasure trove of success in intellectual and professional pursuits, he had a really thriving personal life in terms of his enthusiasm and passions for other things beyond his work. A really tremendous scholar, and I'm very fortunate to have been trained by him in part and to have had the opportunity to work with him and the others there on the team. And the truth is, there's much more to his career, his life, and his legacy than the things that I've said and that we've been able to share. Time limits us from doing that. And I think if you can imagine all the connections that are consequent upon his work and the people that he has worked with and the people that he has trained, you can see like really how incredible his legacy really is. I could kind of cool down a little bit at a personal level because many of these things you'll hear over and over. But at a personal level, what was really impactful for me, I would say these are the three things that I learned from Prof. I think to say that Prof was proud of Neil would be a real understatement. And I think he really interpreted children as a joy. I would say professionally, he really focused and emphasized on creative mastery, as Dr. M mentioned, being meticulous. And he was really passionate about adding international flavor to work, collaborating with people from around the world. The world is a big place with lots of opportunities to work with people, to advance science, and to help to address the challenges that face patients, as Dr. Horsberg mentioned. And so, as things have turned out, we've had children of our own, and so Denise and I are really happy to be parents of these three children here. And when we were in Jamaica, got the opportunity to visit the university chapel where we got married with all three of them. And this is them on the first day of school about 20 days ago. And I think Prof is right that children really are an absolute joy. And with regard to working in international flavor, I've lifted this photo from Mayo Clinic. Uh, they kind of capture the faculty networks, and so this is the network that they've captured for me, and I've enjoyed the opportunity to collaborate with people. So I'm working on the international flavor, like Prof said, and you and the audience will be the judge of how I'm doing on working on mastery. I think that's going to be a lifelong process, but hopefully I'm making steps that Prof will be happy with. I would say if I could try to reduce all of this in terms of what Prof really contributed or what he taught will be in this kind of interaction we had shortly before I left Jamaica in 2009. And the conversation went something like, when you get to Mayo, and he went on a little bit, he said, you should do, and good things will happen. And the content of it doesn't matter so much. I think one of the really crucial things was that he cast a vision for me, some specific goals that he thought that I could accomplish, some specific things he thought that I could do that would make a difference. And I think that that's a very great gift that you as faculty could give to trainees, or you who are parents could give to your children, to cast a vision for them of the things that would be and the things that could be, if they put in the effort of the opportunities that are there for them to make a contribution, or for that contribution to make a difference. And that's something that I found to be very motivated, and I've navigated these years of my career, and I'm really appreciative of this particular gift. And I think many of the things he did constantly cast a vision of what was possible, with the effort, with the work, with provided that you have those opportunities. And of course, he would not just give words, but you could see from the email with Neil, he was definitely follow up his words with action. So today I'm going to focus on molecular discoveries that have changed liver tumor pathology. This is the second portion of the presentation. I'm going to illustrate this predominantly using the example of fibrolamellar carcinoma, which I've worked on a lot. Prof. Hanchard and I talked about this tumor type a lot casually in his office. I remember room 16, I think it was. And I'm briefly going to include some other examples from some other tumors that I have been working on, and how molecular discoveries have altered diagnostics, clinical care, and research with regard to each of these examples. So I have no financial conflicts of interest to disclose relative to this presentation. And I'm confident that if you're able to remain engaged through the remainder of this presentation, you'll be able to describe the genomic basis for fibrolamellar carcinoma. You'll be able to discuss the optimal approach to making that diagnosis. And you'll be able to discuss molecular tools in the assessment of the biology of liver tumors. So I think a pretty ambitious agenda, but I'm confident in you that you'll be able to do this. So fibrolamellar carcinoma was originally recognized by Edmondson in 1956. This is the original photomicrograph published in that manuscript, and this well, no name was given for this tumor, but this is the original photomicrograph. And it wasn't until John Craig and colleagues wrote a series in 1980 that this name fibrolamellar carcinoma was actually chosen for this tumor. And in that paper, John Craig and colleagues refined the clinical and histologic features of the tumor, characterizing it as a tumor that arose in young individuals without underlying cirrhosis and was characterized by these histologic features: tumor cells with abundant eosinophilic cytoplasm, separated by rivers of fibrosis, with nuclei that featured open chromatin, prominent macronucleoli, and it could have intracytoplasmic steatosis, which is, the Latin derivation of fat, and also could have these pale bodies. And on higher magnification, I want to illustrate that the nuclei are really characteristic. You can see there's open chromatin, prominent nucleoli, a real characteristic feature of fibrolamellar carcinoma. And largely, once that discovery was made in 1956 by Emerson, not much change in the diagnosis and the care of patients with fibrolamellar carcinoma for 50 years. It was characterized as a unique primary liver tumor. It was believed to be a variant of hepatocellular carcinoma, so it was just kind of a type in young people without chronic liver disease, and there was a thought that it was possibly had a better prognosis than conventional hepatocellular carcinoma. Now, then some controversy began to emerge. There were reports of cases of fibrolamellar carcinoma in older patients, reports of fibrolamellar carcinoma in patients with cirrhosis, in patients with elevated alpha-fetoprotein, the dubious entity of mixed fibrolamellar carcinoma, hepatocellular carcinoma, and there continued to be controversy regarding the prognosis of fibrolamellar carcinoma. So in that 50 years, not much has changed, but there were a lot of things that people had doubts about. And the root of much of this controversy really is one thing: how do you diagnose fibrolamellar carcinoma? If you had a real robust criteria, or a real robust set of criteria, you would be able to determine, well, these patients with cirrhosis don't have fibrolamellar carcinoma, or they do. And I think that was the real root of the issue. How do you actually make this diagnosis? In the interim, we also noted that fibrolamellar carcinoma is positive for keratin 7. You can see the HD photomicrograph here. The pathologist here will see that the cells are decorated by keratin 7, and they are positive for CD68 because of the prominent mitochondrial burden in these tumor cells, and so picking up this data from the lysosomal marker CD68. But a major breakthrough happened in 2014. I remember when this came out. I remember this paper came out on a Friday in 2014. I was taking a flight to a meeting on the Saturday. And this was a paper wherein Sandy Simon's group, written by Honeyman, and included Sandy Simon's daughter, who is a survivor of fibrolamellar carcinoma, detected a recurrent fusion gene, DNA JB1 PRKACA, in fibrolamellar carcinoma. And that dramatically transformed the field in the subsequent years, because now we had a robust diagnostic biomarker for fibrolamellar carcinoma. And so what they found was this: on chromosome 19, on the short arm of chromosome 19, the two genes, the NAJB1 and PRKACA, which are normally arranged in this particular manner, with 400 kilobases of DNA between them, there was this heterozygous deletion that brought the NAJB1 infused it with PRKACA in frame, giving rise to a functional protein, or certainly a protein that can be transcribed. And they were able to show in their study that it was a functional protein. And they found this fusion transcript in all of the fibrolamellar carcinomas they tested initially. And so this was an important discovery. Because we were interested in this, and we had actually obtained the funding to actually do the experiment they did, but they beat us to the chase. We followed up with our study showing that not only was this a sensitive finding, which they reported, but this was a specific finding. We looked at a range of other liver tumors, including hepatoblastoma, conventional hepatocellular carcinoma, cholangiocarcinoma, and we didn't find this fusion gene in any of them. And so we reported this out as a specific finding of fibrolamellar carcinoma to pave the way for this to be a test that will be used diagnostically. In concert with that, we developed a FISH assay, and this is a slide to illustrate how that FISH assay would work. On the short arm of chromosome 19, that's where the fusion gene is located. And so in normal chromosome 19, what you would expect to see in the nuclei here in interphase will be two intact yellow signals, because of the overlapping red and green signals. And in a case of fibrolamellar carcinoma with the rearrangement, you'd have a separate green and an intact yellow, which is what you see also here in interphase: separate green, intact yellow. And that would be the abnormal signal pattern that would indicate a rearrangement of that locus and support the interpretation of a fibrolamellar carcinoma. So now we had a novel diagnostic test. We also looked at the stroma, those characteristic bands of fibrosis. And even though there are some fibroblasts in there, those fibroblasts are non-neoplastic. And using this discovery and this particular tool, we started to dig into the tumor biology of fibrolamellar carcinoma. And so we embarked on a study with a large group of really well-known liver pathologists from around the world to see what's the role of molecular testing in the clinical diagnosis of fibrolamellar carcinoma. And so this was a list of really well-known experts. You can see the centers that came from, shown here on the map, some from the Middle East or Turkey, which is kind of on the border of Europe and Asia, some from Europe, and then it's spread across the United States and Brazil. The majority of the cases, about 40% of the cases, came from here, Mayo and Rochester. And we also obtained a TMA from colleagues in Germany, which allowed us to study additional cases, as well as some useful controls, conventional hepatocellular carcinoma, fatty liver, normal liver. Now, I want to emphasize that these cases were submitted by excellent pathologists with significant experience. In fact, I was the least experienced of all the people who were involved in this study. And this was an example of one of the cases that was submitted. This is actually two cases that were submitted. You can see the characteristic morphology. These are kind of ribbons of fibrosis. They divide the tumor cells into trabecula. You can see the nuclear features are the same here, and also in the alcohol fixed preparation. And again, in another example, the ribbons of fibrosis, nest of tumor cells, abundant eosinophilic cytoplasm. And when we looked at interphase fish, you can see separate green signals, intact yellow signals. So it's a typical fibrolamellar carcinoma. Many of the cases fit this particular description in the study. And here again, showing you perhaps in a larger view, separate green, intact yellow. That's the pattern that we're looking for. And you can see this repeatedly in cases. Well, one of the interesting things that we observed as we went through testing these cases from these expert pathologists is that 9% of the submitted fibrolamellar carcinomas, they were not fibrolamellar carcinoma. So almost 10% of the time the experts were wrong, even though these are cases that were diagnosed, they're re-reviewed, and then they sent them to us, at least not by that gold standard. And I reflected on this a little bit and looked back into the literature, and here was a very interesting study that should have reassured us that, you know what, I guess it depends on how you look at it. It should have reassured us that we were on the right path, but it also should have challenged us that this is something we should expect. You can see that this study was offered by some really well-known people in liver pathology, Zach Goodman, Linda Farrell, Gabriel Malou, Stefan Humscher. This is like the who's who of liver pathology on this particular paper. They asked the question if the histological diagnosis of primary liver carcinomas is reproducible among experts. And when they looked at these 25 cases, you can see for these kappas here that it was really modest. In fact, these expert liver pathologists really could not agree on how to diagnose most of these 25 cases. And you can see here from the histogram that the agreement is not very good. There's some liver pathologists who issued largely one diagnosis. This person only had two choices that they made across the 25 cases, but you can see the others all had a variety of different frequencies for the various diagnoses. What's perhaps even more troubling is that in about 70% of cases, these are the cases where they were most confident. They don't want any stays or special stays to help make an interpretation. Their standardized kappa was even lower. So the cases they were most confident about, they agreed on less. And the diagnosis of fibrolamellar carcinoma was offered many more times than it actually was made in these particular cases, suggesting that this is actually a problem for expert pathologists. And so you might say, well, Rhonda, that was 2009. This is 2021. In 2017, this is a study looking at the Cancer Genome Atlas, which is a large effort to understand many aspects of cancer. And expert pathologists, again, reviewing cases of fibrolamellar carcinoma in only a small number of cases, now only 6 examples. These experts were only able to get the diagnosis correct in 3 of those 6 cases, suggesting that, expert pathologists really continue to have a challenge making this diagnosis, even though it's been known for about 50 years what this diagnosis is. I think making the diagnosis is challenging. And when they were aware of the results of the fusion testing, they were able to make this diagnosis accurately. But the challenge is not unique to academic pathologists alone. Even community pathologists, people practicing routinely in clinical practice, actually are struggling and have struggled with making this diagnosis. In this study based on the SEER database, which basically pulls the diagnoses made in the community over a large period of time, largely to look for at the epidemiology of disease, a large and well-known and often cited study, you can see that the ages of patients with fibrolamellar carcinoma, including patients over 60 years of age at the first time of diagnosis, in about 1/5 of cases. So while we all know that fibrolamellar carcinoma rate is a disease of the young, it suggests that in the field, in the community, there's a struggle with making an accurate diagnosis. And we know immunohistochemistry is helpful. Carotid 7 and CD68, as I showed you, can be positive in cases, but they may be patchy or negative, particularly when we're reviewing biopsies, where you have a small portion of tissue. We know a significant minority of hepatocellular carcinomas express carotid 7, and you can see co-expression of carotid 7 and CD68 in conventional hepatocellular carcinomas. That has been something that we've also published in our group. But when we look at the results of molecular testing across that study, where we were working with this kind of multi-institutional, international group of pathologists, we were able to make the diagnosis accurately in virtually all cases. There was one case that stood out, a patient whose mother had the cardiac complex. If you could stick a mental pin there, we'll revisit that later. And we found this fusion gene both in primaries and in metastases. And so we really believe that this is now a better gold standard for making the diagnosis of fibrolamellar carcinoma in the appropriate context. We looked at all those controls which we collected, and all the control cases were all negative, which was appropriate. And of course, the same thing is true for the normal tissues as well. And so we turned our attention to this issue of the so-called mixed fibrolamellar carcinoma and hepatocellular carcinoma. And this is a case from Mayo Clinic here in Rochester that actually was in the TCGA and originally misclassified there. You can see there are some areas here that just look like common fibrolamellar carcinoma that you would expect. And then there's some areas here that just look like something different. But if you look at these at the cytogenetic level, they're the same. And we were able to do some follow-up studies on some other quote unquote weird looking fibrolamellar carcinomas or fibrolamellar carcinomas with weird areas. And I think the truth is these are not mixed fibrolamellar carcinoma HCC. These are just fibrolamellar carcinomas with some unusual areas. And I think this discovery of this fusion gene has helped to ameliorate that particular source of confusion in the field. So once we were able to detect this fusion gene, we were able to improve diagnosis of fibrolamellar carcinoma, appreciate the range of morphologic appearances of this tumor type. It's about 100% specific, but of course in context. One of the challenges we face in today's world is people taking things out of context. Out of context, things are very different. It's really important to understand the utility of tests. When you're asking this specific question of a liver tumor that has hepatocellular differentiation, this test is 100% specific. And up to this point, aside from that patient who was the offspring of a woman with the cardiac complex, it was very, very sensitive. It was almost perfectly sensitive. And we validated this in alcohol fixed preparations. By some studies that we did in the lab, we're able to show that this test is informative once we had as few as 50 cells. And of course, this method, while FISH is a great method, there are other methods that work and they have a similar analytic performance. So at this point, I want to say that we were able to summarize that fibrolamellar carcinoma is a distinct morphologic entity with specific molecular biology correlates. And it opened the question, in my mind at least, as to whether or not there's a genotype-phenotype correlation in hepatocellular neoplasia. And I want to kind of pause here to say this is actually what got me interested in pathology, the notion of genotype-phenotype correlation. And that interest was awakened while at the University of West Indies by the faculty there. And that's actually what my career has largely been about. And while we didn't have resources to do a lot of that while I was there in Jamaica, I think they awakened that passion in me and motivated me there individually, the people at the lecturing staff there, but also collectively to pursue a career like this. And so once I got to this juncture in this particular project, this is naturally what I thought about. And so to me, it seemed that it was insufficient to just call all these tumors the same. All HCCs are the same. There is some genetic phenotypic correlation that happens. And so I'm going to take you on a little bit of a detour to look at some other things that are not fibrolamellar carcinoma. But I hope that this detour will be useful to kind of reinforce the point that there's some correlation between what's happening genetically and what we see microscopically. So the first group that I think really brought this out in liver tumor pathology is a French group. And they were the first group to show that if you see a hepatocellular carcinoma with cholestasis, here are these top part of the panel, and you might see this pseudoacinar pattern, that they correspond to groups that have beta-catenin mutation or tumors that have beta-catenin mutations. They were the first group to really put this on the map in 2007. In 2017, a friend of mine, Julia Calderaro, also from France, separately was able to show that there is a funny kind of HCC, which he coined this name macular trabecular massive. You know, the French just have a great way of saying everything. I like that name. Macular trabecular massive, which describes a tumor that has these massive trabeculae and tend to form large tumors, but not just morphologically distinctive. He was able to show from their group in Paris that there were also some distinctive correlates of that. They had a high alpha fetoprotein, frequently had viral hepatitis B, satellite nodules, extensive vascular invasion, and then some other molecular correlates. And he confirmed these findings largely in the TCGA, so in a separate cohort, to show that this was something that was being seen reproducibly across the cohorts. And in subsequent studies, he showed that this is a diagnosis that pathologists can make reproducibly. In addition, he also showed in that paper that macrotrabecular massive hepatocellular carcinoma has a worse prognosis than conventional HCC, shown both in the Paris group and also in the TCGA. And so I think when we think about hepatocellular carcinoma, we need to consider that they're all probably not the same, but we need to reevaluate how we look at the morphology, as we did for fibrolamellar carcinoma, how Julian and his colleagues are doing for macrotrabecular massive, and correlate that with molecular and other tools, and we may I guess, derive new knowledge that helps us to approach HCC in a different way. So HCC or hepatocellular carcinoma, fibrolamellar carcinoma are not the only liver tumors that have been affected by these advances. There are three others that I want to touch on as we proceed through this portion of the presentation, where molecular observations have opened our eyes to some new vistas in these particular tumors. So the first is epithelioid hemangioendothelioma. This is a tumor described by Enzinger and Weiss, a great duo of pathologists, giants in the field. And what they reported was a tumor that's really characterized by its matrix. It has a really distinctive matrix that's really kind of chalky and mixoid. It has really distinctive cells and a propensity to show vascular invasion. It mimics adenocarcinoma frequently, and that's one of the main misdiagnoses that is made, and it's a tumor with a varied prognosis. Almost simultaneously in 2011, two groups discovered a recurrent fusion gene in this particular tumor type, which again is a similar pattern to what you saw in fibrolamellar carcinoma. It triggered a new wave of scholarship. And so the ensuing wave of scholarship allowed us to appreciate more about the morphologic spectrum. And one of the things we also discovered is that we had a tumor also now with a distinctive matrix, a little bit calcified. But in this example, you can appreciate that this tumor is very vasoformative. You can see vessels actually being formed here. The pathologists in the audience will recognize this readily. But on high magnification, the cells have this abundant cytoplasm. I'm a big cytoplasm kind of guy. So there's this abundant cytoplasm and a histiocytoid kind of look. These tumors now are characterized by YAP1 TFE3 fusion. So a different fusion gene. So we appreciated that this distinctive morphology or difference in morphology corresponds to a different molecular event. And what we believe now, or what we certainly understand from the available data now, is that this variant appears to have a better prognosis, and so the fusion gene likely matters. And so making this morphologic distinction and recognizing the different molecular correlate likely has some significance relative to the outcome of the patient. And another tumor that will be intrahepatic cholangiocarcinoma, something that I've really enjoyed working on. We were a part of groups making the report in 2014 that a subset of them harbor FGFR2 fusions, which we believe, or at the time, we believe that the time subsequently has been confirmed, are targetable alterations. And in the ensuing period, the FDA has approved 2 agents to target FGFR2 fusions in intrahepatic cholangiocarcinomas. There are multiple trials that have shown that they contribute to controlling the disease. And so detection of these fusions is clinically relevant. And this is something that we do routinely in our practice here in Rochester, and I'm sure happens in practices across the US, Europe, and in Asia. And the third tumor that I will kind of introduce to you is the mucinous cystic neoplasm of the liver, which is like something that we're currently working on. This is a tumor that is in the liver, in the pancreas. Sometimes you can see it in the mesentery, which has a stroma that resembles the normal ovary, but except it's not in the ovary. It has this characteristic epithelium overlying it. And in working on this tumor, we did a variety of things, but one of which we realized that it doesn't just look like ovary. We were kind of digging a little bit deeper. We realized that this tumor not only had these ovarian markers, which many people know, it expressed progesterone and estrogen receptors, but one of the interesting things is that it had sex hormone stromal markers. So that was interesting. This led us to ask the question, what about markers of hormone biosynthesis? And indeed, we found at RNA-seq at transcriptomic levels that there were markers of uh, sex hormone biosynthesis, suggested that these tumors, the stroma doesn't only look like the ovary, but is actually producing hormones, or capable of producing hormones, as will be the case in the ovary. And we follow this up with immunohistochemistry to validate it, and is an ongoing aspect of our work. So at this point, I would like to say that molecular discoveries, what they're doing is bringing the picture of liver tumors into sharper focus. And at this point, this is something that Prof would always say. He had this tendency of like stretching out my name. He would say Rondell, which I always thought was hilarious. I don't know why he did that, but I noticed that other people in the pathology department there in Jamaica would do that. So like Dr. N would often say things like that. And we would actually refer to Dr. N as Dr. N. I don't know why we did that, but it always kind of signaled when something was going to change, right? So a change in focus. And so I want to introduce you to a detour back to the patient whose mother had the cardiac complex. I'm going to skip over some biochemistry, and hopefully there are not too many biochemists on the call. But what we were interested in is whether the PRKAR1A gene, which is encodes the regulatory subunit, which would be almost, if you could imagine, the antagonist to PRKACA in a physiologic state. We were interested if that gene, which are the mutations in which underlie the cardiac complex, could be involved in fibrolamellar carcinoma in patients with the cardiac complex. And so we went about reviewing liver tumors for patients with the cardiac complex. That was a ton of fun. We looked for some sporadic fibrolamellar carcinomas as controls. And what we found in patients with the cardiac complex is that we found some fibrolamellar carcinomas from Mexico, from Europe. So we found them, they looked like it, the immunohistochemistry fit, but they didn't have the fusion gene. Instead, what they had was a loss of PRKAR1A. And we followed this up with molecular testing to confirm that, yes, they did have loss of PRKAR1A, one germline hit and one subsequent loss in the tumor. And so these tumors could be driven not only by this gain of function fusion gene, which I mentioned earlier, but also because of loss of function, as in the example of patients with the cardiac complex. And here you can see what the normal should look like, or the quote unquote sporadic case should look like. And we also looked in the literature. We found this report in 1987. We were unable to retrieve the tumor because the materials were discarded when we were trying to find it in 2018, which was about 30 years after this case had happened. But in this particular kindred, this particular individual had a liver tumor that was possibly fibrolamellar carcinoma, and another independent example of what we were reporting. So it was an independent observation. The interesting thing that we also concluded in our paper, when you look at the number of people in this family and the number of patients in cardi families, it doesn't appear that all of them will develop fibrolamellar carcinoma, suggesting that there's variable expressivity of the liver manifestation in patients and with the cardiac complex. So the interesting thing about fibrolamellar carcinoma in individuals with the cardiac complex is that there's an absence of the classic fusion gene. There's a loss of expression of the PRKAR1A protein, which you would expect, and that corresponds to inactivated mutations. And then we suggested that individuals with the complex should be considered for screening for fibrolamellar carcinoma to ensure we can detect this tumor early. And the other interesting thing that came up as we worked on this is that this is something that could potentially happen in a sporadic setting, because in one of our patients, this particular patient from Europe, there was no known family history, and he developed fibrolamellar carcinoma at an older age, but we were unable to track him down for further follow-up. So at this point, what we had kind of summarized is that fibrolamellar carcinoma displays distinctive morphology, distinctive clinical features, distinctive immunophenotype. And if you could draw this as a diagram, this would all be boiled out to protein kinase A activation, both because of a gain of function mutation, and then another component because of a loss of function. one of the interesting things that happens is that when you think you have things all figured out, you don't. There's more to understand, and that actually happened in this particular case. I remember the specific day, this specific case. This is a patient uh, case that we receive in our particular practice, a young boy with a liver mass. And in this small biopsy, both characters 7 and CD68 were negative. And this is one of the reasons why I'm cautious about relying too much on immunohistochemistry for this diagnosis. We had done some mass spectrometry-based proteomics on a research basis, and we saw some features that suggested that this was actually a fibroblastoid carcinoma. And to me, morphologically, it actually looks like it. The distinctive thing here being that you have the right cytology, you don't have the fibrous mass, but again, this is a small biopsy. When we did fish, what we actually saw was this, these clouds of green. So not separate green, but clouds of green. And this was indeed a case of amplification of PRKACA. Rather than a gain of function fusion gene, you just had gated copies of PRKACA, almost analogous to what you would see in a case of with HER2 in terms of seeing the cloud of signals. That's just kind of a rough analogy. But PRKACA amplification, though, underlying fibroblastoid carcinoma. And so we switched from our Venn diagram to kind of show in this particular format, but protease kinase A activation seems to be the key thing, the crucial thing, mostly because of a gain in function fusion, rarely because of a loss of function mutation, usually when one germline hit, but and rarely in individuals you may see amplification. Now, why does this all matter? I think if you look at this as diagnostically useful, but one of the other interesting things is if you look at the AHSCC stage, the system which we use to stage liver carcinomas, and we do use for fibroblastoid carcinoma, it doesn't stratify fibroblastoid carcinoma as well. And I think that's because the biology of fibroblastoid carcinomas is different, and the natural history of fibroblastoid carcinoma is different from hepatocellular carcinoma. And this table kind of summarizes some of these differences. Fibroblastoid carcinoma occurs in young people without cirrhosis, without alpha fetoprotein elevation. It frequently involves regional lymph nodes, unlike conventional hepatocellular carcinoma. It frequently spreads to the peritoneum and will recur late, whereas conventional hepatocellular carcinoma recurs early. It has a distinctive proteome, which I did talk very much about, something that we kind of use in research setting, and it is characterized by protease kinase A abnormalities, which is different from what you see in conventional hepatocellular carcinoma. Now, one of the things that I think I also learned from Prof is that time goes by quickly. That's something that we're learning and cherishing with our own children. These are our children a couple of years ago. So I was just in the beginning of the talk looking at them at the first day of school, but I remember these days just like they were yesterday. And believe me, the time goes by quickly. It's really important that you enjoy it. And I would like to conclude the presentation by wrapping up on my objectives, which is to show that fibroblastoid carcinoma is a distinctive tumor, it has distinctive morphology and clinical features, a distinctive transcriptome at the RNA level, protein level, and genomic level. Is driven by protein kinase A abnormalities, most often DNA JB1, PRKACA fusion gene. And using this just as a large example, there are other examples that I highlighted on some very briefly in the presentation where molecular tools and molecular discoveries are transforming the way we think about making a diagnosis. For example, in the case of epithelial hemangioendothelioma, the way that we care for patients in the context of FGFR2 fusions and intrahepatic cholangiocarcinoma, and the way we're evaluating tumor biology and the way that we're looking at mucin-assisted neoplasms to understand early stages of cholangiocarcinoma. So with that, I'd like to thank you for your time and attention, and I'm available to answer any questions you might have. Karen, I think you're on mute.
Sorry. Thank you so much, Dr. Graham, for that wonderful presentation. If I start with the way you really put into perspective Prof's wide reach within the practice of pathology, then medicine, then life, it was actually a bit moving to hear you talk about how he influenced our lives and more specifically yours. And then to go on to talk about a very difficult topic. A lot of us get apprehensive when we start talking about genetics, but like last year, when Neil took something very complex and made it easier to understand, I think you did the same for us this year. So we have a few questions. The first question is from Dr. Ravi Saw, and I don't know if Dr. Graham knows Dr. Saw, and I hope Dr. Saw doesn't mind that I say no. Dr. Saw is a graduate from the pathology residency program here, who also migrated to the US, completed post-doctoral training and practices in the US now. So Dr. Saw's question, Dr. Graham, is in the study where expert pathologists differed in classifying 25 hepatocellular carcinoma cases, how were the diagnoses made? Was it all FNA, core biopsies, resections, or an admixture? Because he's wondering if sample size presented, the limitation in sample size influenced the interpretation.
So that's a great question, and I'm pleased to electronically meet Dr. Saw, as it were. I've never met him in person, but I have heard of him, and he mentioned in the chat that he is tuning in from Oklahoma City, and to his point, the samples were all histology samples, so no cytology samples in that particular study, but some. You know, there are different sizes, and sampling always does play a difference or play a role in the differences that the differences in interpretations, but all 17 pathologists had the same sample. So they would have all faced the same limitations. So that, if that could address his question. And I see some other questions in the chat, which I could go ahead and ask if you want me to. They're kind of related. So Dr. Warm, who also participated in my training, and I'm fortunate to have been trained by her, was asking the question is, how does this translate into treatment? One of the unfortunate things with hepatocellular carcinoma in general, and familial carcinoma in particular, is that there is no real great systemic treatment. But what is really well recognized now is that familial carcinoma can benefit from metastasectomy and ongoing resections, even from widely metastatic disease. If you can, the disease burden can be controlled surgically. Patients respond to that. In contrast, for conventional hepatocellular carcinomas, that's not the case. So there is an influence in how the surgeons will perceive in cases like that. And I see another question for Dr. Baker, Dr. Akil Baker. So that's a good question. What do we use as our gold standard for the fusion test is a really good question. So to establish a new gold standard, what do we use as our initial standard? So we use the impression of all of us who were participating in that study at the time. We combined it with the immunostain data, and we also looked at mass spectrometry-based proteomics. We took some classic examples. We looked at mass spectrometry-based proteomics where we looked at a large percentage of the proteome, more than 3,000 different proteins, and we looked at what if we saw consistent signatures. And there were some proteins we saw consistently detected if cases that were looked like classic garden variety familial carcinomas. And we then went backward now and looked at cases that had all those features. We focused on the nuclei, and we realized that there were a lot of cases that we interpreted as familial carcinoma, but there were things that suggested that that probably wasn't the best diagnosis. But those fine histologic interpretations, that's the challenge. And on needle biopsies, that's a major challenge. And so this was the advantage of the molecular testing, particularly on small samples. So I hope that was helpful, Akil. I can elaborate more if need be. I see Neil has his hand raised.
Actually, so I had sort of two questions. One is sort of in follow-up with what Dr. Worf was suggesting. So, like, there's been all the success with DVEC, right? For BCL-ABL, then sort of fusion gene is sort of fusion gene treatment, something on the horizon for these kind of hepatocellular carcinomas if you happen to catch them early enough, right?
It's a good question. I know there are groups working on that, like Sandy Simon's group is working on it, and we have a research group, a liver spore here, where they're working on seeing whether they can target the fusion. If they can, there's some complexity with developing the chimera, and then protein kinase is involved in many functions, so they have to be cognizant of the off-target effects, which are going to could be substantial if the targeting isn't great, but that certainly is an ongoing direction.
And my other question was, so, you talked about this fibrolamellar carcinoma that sort of looked a little bit unusual. And I just wondered whether or not that has, like, if you sequence across the actual fusion, do you see differences either at the sort of variant level or, the size of the deletion or something like that, a different kind of fusion?
It's a super great question. I haven't followed up on that, but I think it is a really relevant question. The idea being, if I interpret you correctly, are these weird areas, these unusual areas, perhaps these areas with increased nuclear to cytoplasmic ratio, do they correspond to other genetic events, secondary events, or maybe there's a greater tumor mutation burden, etc.? And the answer to the question, I don't know. We haven't done that yet. But I think that that's an important future direction. One of the things we're working on now is looking at how has this discovery influenced practice, looking across our reference practice and working with others in different places to see how they actually make the diagnosis now of fibrolamellar carcinoma, to perhaps the point made by Dr. Horsmer earlier, to see what's the most efficient way to make the diagnosis, the most cost-effective way to make the diagnosis.
Thanks.
And look, I see another question from Ravi. Karen, can I go ahead and answer that?
Yes, please. I was about to point it out.
Okay, so Ravi saw us. Hopefully, I don't know if others can see the questions.
No,
but
you have to read the questions, please.
Okay, so I'll read the Ravi's question, which is: should molecular testing be performed on all intrahepatic cholangiocarcinomas looking for FGFR2 fusions? It's a really good question. I don't know I would say I don't think the answer is all. I think the answer will be probably those patients who are not amenable to surgical cure, where there's the option to receive FGFR2 inhibitor therapy. So if the option to receive that therapy exists, I think it would be a reasonable thing to do. In our practice, we're actually in the process of designing clinical decision support. So when we make a diagnosis in pathology, there is a logic in the computer system to identify that diagnosis, and it matches to our test menu to say this is a patient potentially who could get this, and then based on some other clinical factors will determine whether the order is instituted or not. So I wouldn't say for all cases, for routine clinical care, because it's unlikely to be cost effective. Thank you. Thanks for that question.
Okay, if we have no further questions, thanks again, Dr. Graham, for your excellent presentation. I invite you to read the comments in the comments box while I ask Dr. Sherry-Ann Spencer, who is a lecturer in our Department of Pathology here at University of the West Indies, to please make closing remarks.
Good evening, everyone. Thank you, Dr. Bishop. Ladies and gentlemen, we have reached the end of our third annual memorial lecture honoring Professor Barrie Hanchard, who has made such an indelible impression on us all. It is such a pleasure for me to express heartfelt gratitude to everyone involved in making this evening a success, including the organizers of this event, Mr. Hanif Smith and his team at the Strategic Management and Training Consultants Career Institute. They created the virtual program and they made it a reality, and they took us smoothly through this evening. More specifically, gratitude is extended to our moderator, Dr. Karen Bishop, who skillfully presided over this evening's program. Our head of department, Dr. Tracy Gibson, who greeted us with warmth and cheer. Dr. Susan Shirley, thank you for blessing this evening's proceedings with opening prayer. I am certain you will all agree with me that we are truly appreciative of the greetings received from Dr. Carl Bruce, Professor Joseph Plummer, and Dr. Maria Clara Horsberg. Thank you, Dr. Nadia Williams. You so eloquently delivered a riveting biography. Special thanks to Dr. Neil Hanchard, who graciously introduced our guest speaker. Dr. Rondell Graham, we are appreciative of the artful and articulate manner in which you delivered your lecture on lessons in life and liver pathology. It was both informative and very interesting. Thank you for obliging us with the snippets into what life as a pathology resident with Prof was like and how the experience has had such great influence on you. A big thank you to the organizing committee, as well as Dr. Brooke Harrison, who created the virtual invitations and a program. And thank you to Ms. Hyper Nelson and Mrs. Karen Black-Grant for assisting with the dissemination of same. I would also like to thank this evening's sponsors, Roche Pharma and Diagnostics. And I would be remiss if I did not thank you, the audience, for attending, celebrating, and memorializing Professor Hanchard with us this evening. This has been a truly wonderful program. I will now hand over to Dr. Bishop.
Thank you, Dr. Spencer. Just want to echo your remarks, thanking Roche in particular for sponsoring this evening's proceedings. That really helped us with allowing a large number of people to join us in this virtual meeting. Thanks again, Dr. Graham, and to everyone. And as always, thanks to the Hanchard and Thomas family, especially Dr. Thomas and Neil, for giving us their blessing every year to continue to honor Prof. So with that, we wish you a good evening and ask you to look forward to hopefully the 4th memorial lecture honoring Professor Barrie Hanchard. Good evening to you all. Is Rondell there still?
Yes, I am.
Thanks again, Rondell.
Thanks, Karen. I had a fun time. Thanks for inviting me. This is super terrific.
Okay.
We'll talk some more soon.
Everyone. Thanks, Neil.
I hope you saw one of the comments from Carl Bruce, who I expect thinks that COVID will be over soon. He's looking forward to you participating in person in the future.
Yeah, well, yeah, if there's anything I can do to help, let me know. I really enjoyed this. This was a ton of fun for me, and it brought back a lot of great memories.
It was obvious that you were having a great time.
Oh, yeah,
it's always a blessing to see somebody who loves what they're doing.
Yeah. Yeah, we'll have to connect again soon. For whatever reason, my video won't start back. The host has kind of stopped my video, but maybe that's for the best.
It's not you alone, Dr. Graham. I think he has blocked off a few of us.
Oh, he's probably cutting us off.
There you are. There you are.
There
you are.
Okay.
I think our conversation is open to the audience as well, though. So
Yes.
just to be
Is
aware.
it? I thought they were gone.
I think so. Yeah.
No, that's fine.
I think that's
Yeah. Super
Okay,
good.
everyone.
Take care, everyone. Hope you have a good
Neil.
evening.
All right, be safe.
We'll
all.
talk soon about next
Be
year,
safe.
of course.
Absolutely.
Okay, then.
Hopefully.
Yes.
Yeah,
Okay,
I'll-
bye.
Cheers.
Bye.
Person.
Bye all.
Bye.